One thing we'll want to talk about is that in human brains, when they reach about an age of 80, usually lose about 5% of their weight. Some of this might be due to problems in degenerative disorders like Alzheimer's, but do neurons die off just as part of a normal course of life or normal aging? They actually do. You can see that the lateral prefrontal cortex starts to decline in volume reduction, as seen here, but in other parts of the brain, it remains pretty stable. Here you can see the volume of the brain region, and then down here, the age going up to 80. This first one shows you the volume is lost, but the second one for the primary visual cortex, as an example, doesn't go down as much. This is an image of the prefrontal neuron from a young monkey and shows a close-up here showing the difference between a young monkey on the left and an aged monkey on the right. On both images, that the protrusions are not as great as the one in the young monkey. Some researchers, such as Carol Barnes and her colleagues, have suggested that the number of neurons doesn't decline totally with aging, but what does change is the ability to change synapse strength. Remember, long-term potentiation is the process where activity of two neurons strengthens the synapse between them. This is how neurons encode new learning. If LTP occurs but fades away with new learning, then the learning will be lost. Barnes and colleagues placed rats in a maze and examined their hippocampus place cells. Remember, place cells are cells that fire when a rat is in a particular location on a maze. This was discussed in Chapter 3. These images here show activity that was recorded from several such place cells in the hippocampus of young rats and older rats. The different colors indicate where each neuron fired. One neuron, such as the one in blue here, fired when the rat was near a left end of a center arm in the maze. If the rat turned the corner to the right, as you can see, the neuron stopped firing and another neuron would fire, which as you can see merges to the color green. So looking at this, we know which neuron was firing at any given moment and can determine the location of the rat. So session 1 is where the rat was first in the maze, and then session 2 is where it returned to the maze the next day. What do you see when comparing these two differences in the young rat between session 1 and session 2? They're pretty similar, aren't they? Well, the young rat sessions, the place-dependent neural firing was very similar. But with older or aged rats placed in a maze in session 2, which took place on the next day again, the aged rat's firing pattern changed dramatically. This suggests that the spatial learning from the first day in the aged rat did not persist. The ability to learn and recognize the environment, for example, did not last, so learning and remembering how to navigate this environment did not stick. Perhaps, as Carol Barnes and her colleagues noted, a loss of synaptic strength as we age might be the reason for these rats and for humans. The loss of synaptic stability. So yes, the aging brain does lose neurons and synapses, and long-term potentiation may not be as stable, but the brain has shown to grow new neurons. It was actually believed that humans and animals contain all the neurons that they have ever had, but we know that neurogenesis does occur all throughout life, but of course much less in older adults compared to the developing brain when young. Adult neurogenesis was first observed in birds and has since been observed in amphibians, fish, and reptiles. This image here shows that a cell is an anaphase, meaning during a phase of cell division where a cell starts to separate.